Hello students, today we'll be discussing on the topic recombinant DNA technology. Introduction. Recombinant DNA or rDNA is made by combining DNA from two or more sources. The process often involves combining DNA of different organisms. Recombinant DNA technology depends on the ability to cut and rejoin DNA molecules at points which are identified by specific sequences of nucleotide bases called restriction sites. DNA fragments are cut out of their normal position in the chromosome using restriction endonucleases and then inserted into other chromosomes or DNA molecules using enzymes called ligases. The resultant recombined DNA molecule is known as the recombinant DNA. There are various terms and tools that are used in the recombinant DNA technology, some of which are discussed below. First, we'll discuss about complementary DNA. The term cDNA refers to complementary DNA. cDNA is known to be synthesized or manufactured from an mRNA or messenger RNA template. It is synthesized in a reaction that is catalyzed by the reverse transcriptase and DNA polymerase enzyme. It is essential to note that cDNA is usually used to clone eukaryotic genes in prokaryotes. When a protein is being synthesized, a gene's DNA is transcribed into an mRNA, which is then translated into a protein. In order to synthesize a recombinant eukaryotic protein in prokaryotes, only the coding sequence should be provided to the bacterial cell. Therefore, the mRNA is reverse transcribed to make a copy of the gene, which is intron-free mRNA. This is referred as a complementary copy of the mRNA. Though there are numerous processes for synthesizing cDNA, the best way of doing so is by using mature or fully spliced mRNA. This is usually done by using the enzyme reverse transcriptase. This enzyme generates its cDNA by pairing RNA base pair to their DNA complements. In order to have a complete profile of gene expressed in an organism, a cDNA library can be created. A cDNA library is made using mRNA instead of DNA as the starting material. The mRNA can be extracted from cells of specific tissues from the organism of interest, and a double-stranded DNA copy is made from a mRNA. Because of the different starting material, cDNA libraries will have some key distinctions from a genomic library. The clones in a cDNA library is small subset of the organism's total gene complement because the organism will only turn on certain genes in certain cells at certain times. For example, a root cDNA library will be expected to have many clones of genes that would not be found in a leaf cDNA library. A cDNA library made from mRNA isolated from young leaves would also have clones of different genes compared to a cDNA library made from senescing leaf mRNA. Some cDNA gene clones would be found in most cDNA libraries. These would be clones of genes that are expressed in all cells in the plant. Genes are expressed at different levels in cells. Therefore, some genes will have thousands of mRNA copies in a cell, while other genes will have a few mRNA copies. When the mRNA sample is used to make a cDNA library, the library will contain proportionally more copies of clones of some genes compared to others. Apart from cDNA library that has a complete profile of genes that are expressed, a genomic library can also be generated by recombinant DNA technology. In genomic library, cloning DNA gives rise to a collection of recombinant DNA molecules, often in plasmid or phage vectors maintained either in suitable hosts. A collection of such independent clone is called a clone bank or library. The term genomic library is referred to describe a set of clones representing the entire genome of an organism, and the production of such a library is usually the first step in isolating a DNA sequence from an organism's genome. A genomic library is a rich resource as it represents the entire genome of an organism and should contain all the genes and their control sequences. The first consideration in constructing a genomic library is the number of clones required. This depends on a variety of factors, the most obvious one being the size of the genome. Thus, a small genome such as that of E. coli will require fewer clones than a more complex one such as the human genome. The type of vectors to be used also has to be considered, which will determine size of fragments that can be cloned. The genomic library is constructed by restriction digesting the entire genome of an organism and producing recombinant DNA of each fragment. The total collection of fragments are considered as a genomic library of the particular organism. In order to generate this genomic library, we need the enzyme called restriction endonucleases. Restriction enzymes recognizes short DNA sequences and cleaves double-stranded DNA at specific sites within or adjacent to these sequences. Approximately 3,000 restriction enzymes recognizing over 230 different DNA sequences have been discovered. They have been found mostly in bacteria but have been isolated from viruses, archaea, and eukaryotes. It has been estimated that 25% of all bacteria contain at least one restriction enzyme, and as many as 7 have been found in a single species. Now let us try to classify the restriction enzymes. Restriction endonucleases are categorized into one of 4 general groups: Type 1, Type 2, Type 3, and homing endonucleases based on their subunit structure, cofactor requirements, specificity of cleavage, and associated methylase activity. Let us consider discussing about restriction modification system, which is an inherent part of the restriction endonucleases. Restriction endonucleases of type 1, 2, and 3 have companion methylase that recognize the same sequence as the endonuclease and methylate each strand at a specific base and position, resulting in either 4-methylcytosine, 5-methylcytosine, 5-hydroxymethylcytosine, or 6-methyladenine. Once methylated, the host DNA is no longer a substrate for the endonuclease. Hemimethylated DNA, such as after a fresh round of replication, is also protected from digestion. Restriction endonucleases requires recognition of specific sequences. Most restriction endonucleases recognize palindromic or partially palindromic sites. A palindrome is defined as a diet symmetry around an axis. For example, EcoR1 has a GAATTC recognition site, which is a palindrome. In the next subtopic, we'll be discussing about types and general properties of restriction endonucleases. Restriction enzymes are traditionally classified into 4 types on the basis of subunit composition, cleavage position, sequence specificity, and cofactor requirements. However, amino acid sequencing has uncovered extraordinary variety among restriction enzymes and revealed that at the molecular level, there are many more than 4 different types. The type 1 enzyme are complex, multi-subunit, combination, restriction and modification enzymes that cut DNA at random far from their recognition sequences. Originally thought to be rare, we now know from the analysis of sequence genome that they are common. Type 1 enzymes are of considerable biochemical interest, but they have little practical value since they do not produce discrete restriction fragments or distinct gel binding patterns. Type 2 enzymes cut DNA at defined position close or within their recognition sequences. They produce discrete restriction fragments and distinct gel binding patterns, and they are the only classes used in laboratory for routine analysis and gene cloning. Rather than forming a single family of related proteins, type 2 enzymes are a collection of unrelated proteins of many different sorts. Type 2 enzymes frequently differ so completely in amino acid sequences from one another and indeed from every other known protein that they exemplify the class of rapidly evolving proteins that are often indicative of involvement in host-parasite interactions. The most common type 2 enzymes are those like EcoR1, HIN3, and NOT1 that cleaves DNA within their recognition sequences. Enzymes of this kind are principal ones available commercially. Most recognize DNA sequences that are symmetric because they bind to the DNA at homodimers, but a few, such as BBVC1, recognizes CCTCAGC sequence, which is symmetric DNA sequence because they bind at heterodimers. The next most common type 2 enzymes usually referred to as type 2S are those like FOC1 and ALW1 that cleave outside of their recognition sequences to one side. Type 2G restriction enzymes, the third major kind of type 2 enzymes, are large combination restriction and modification enzymes, which are 850 to 1250 amino acids in length, in which the two enzymatic activities reside in the same protein chain. These enzymes cleave outside of their recognition sequence and can be classified as those that recognizes continuous sequences. Type 3 enzymes are also large combination, restriction and modification enzymes. They cleave outside of their recognition sequence and requires two such sequences in opposite orientation within the same DNA molecule to accomplish cleavage. They rarely give complete digest. Type 4 enzymes recognize modified, typically methylated DNA. After discussing restriction digestion, we'll now go to the discussion about restriction analysis. Many of the revolutionary changes that have occurred in biology over the past decade can be attributed directly to the ability to manipulate DNA in definite ways. The major tool for this recombinant DNA technology are restriction endonucleases and enzymes that can wind, cut, mend, repress, unwind, transcribe, and replicate DNA. Restriction enzymes are referred to as molecular scissors of the molecular biologist. Restriction endonucleases enzymes cut DNA at specific nucleotide sequence. A sample of an individual's DNA incubated with restriction enzyme is fragmented to millions of DNA stretches of varying sizes. A DNA sample from a different person would have a different nucleotide sequence and would thus be enzymatically chopped up into a very different collection of fragments. The DNA fragments are separated by agarose gel, electrophoresis, and tagged or stained in some fashion so that they can be visualized and studied. Small fragments are lined up in a column, the largest closest to the beginning of the gel and the smallest at the end of the gel after agarose gel electrophoresis. The size of the fragment is then determined using a standardized DNA ladder of known molecular weight. If a DNA sequence is known and available in the nucleotide databases, then the sequence can also be used to determine possible recognition sites of various restriction endonucleases so far discovered. The process can be achieved by using available online tools that helps in analysis of restriction sites. This particular tool is helpful in that there is no requirement of a physical restriction digestion to determine the size of the digested molecule. The restriction digestion map is accurate, time-saving, as well as convenient to use for analysis of restriction sites on a given sequence. In conclusion, we have learned about some of the important tools and terms as well as few techniques that are used in producing a recombinant DNA molecule. Recombinant DNA technology is a powerful technique that is being employed to produce and improve organisms by generating transgenic forms. However, a great caution should be accompanied with genetic engineering research, and code of ethics should be followed before manipulating an organism's genome.